All right, as we're starting the webinar and everybody is starting to come in the room and join, we'd just like to thank everybody for joining us today for this vet chat here about sudden cardiac death in horses. This is a project that Grayson funded this past year in 2021 through the University of Minnesota with Drs. Molly McCue and CN Durward-Akhurst. I hope I pronounced that, didn't butcher it too much. As you'll see, there's a Q&A button at the bottom of your program on Zoom. If you just use that to ask questions, we'll come back on at the end of the program and answer any of those questions in a certain order and no order what they come in, but as probably of relevance comes in. So without any further ado, I'd like to thank you ladies, both doctors, for coming on and doing this today. It's a great honor and pleasure to not only fund your research, but have you come out here and tell everybody about it. And thank you so much again.
Of course, you're welcome. Thanks so much for having us here. We're really excited to talk about the project that we're working on. And just a way of brief introduction. I'm Molly McCue. I've been at the University of Minnesota for my entire faculty career. I'm a large animal equine internist by training, and then my research focuses in genetics. And one of the really great things about being a researcher, and actually my favorite part, is training people and watching them go on to do great things. And so Sean is a brand new faculty member, just about 8 months into being a grown-up, as she says, at the University of Minnesota. I was very blessed that she's also an equine internist. She trained with us. I was her residency advisor, and then she came to do a PhD with me and stayed after a PhD actually to do a postdoc. And we have a lot going on in cardiac arrhythmias in this work, and it's really Sean's baby. And she now as a grown-up is leading the project. So you're going to hear mostly from her, but it's fun to be here and watch her talk about it, because I've known Sean for a long time now. So the best part of research is the people you get to train. But just by a couple ways of introduction, Sean's going to talk about her whole project, which has quite a lot of breadth and depth, all of which is relevant to racehorses, including parts of the project, sort of the bigger picture project. Obviously, Grayson has given us a lot of support, and there are very clear objectives on that project, but we thought it would be important to talk about what we're doing in this area as a whole, because all the pieces fit together to hopefully come out with solutions for the horse. So I'm going to turn it over to Sean and just smile and nod and be the proud academic mother while she talks about her work, because this is really her brainchild.
Thank you very much, Molly, and thank you as well to obviously the Grayson Jockey Club for funding us and for inviting us to talk with you today. I will get started. So as Molly said, we're talking about the whole project, and really our big goal is to identify horses that are at high risk of arrhythmias and sudden cardiac death, with the goal of identifying the ones that are important so we can monitor them more thoroughly and try and reduce the rate of sudden cardiac death. So today I'm going to give a little bit of a review of the equine heart, and probably part of the reason why we see quite as many arrhythmias as we do. And then I'll break it into the next two sections of what we know about arrhythmias, and then what we're doing to try and answer some key questions that I will pose. And then similarly with sudden cardiac death, what we know about sudden cardiac death, and how we're planning on answering some key questions that will hopefully enable us to reduce the rate of sudden cardiac death. Excuse me. So I'll start with a review of the equine heart. And so we all know that horses are absolutely tremendous athletes. But what is really amazing is that even the average thoroughbred racehorse, really, their cardiac potential dwarfs the ability of the very best human sprinters in the world. And this is not, what's interesting is the heart rate is actually relatively similar, but the cardiac output can be 5 times as much as that of humans, and their aerobic capacity is really, really quite impressive. However, when I was looking this up and looking to see whether the horse was the most efficient animal that we could find, I discovered this. This is an Etruscan shrew, and their aerobic capacity is 400 mils per kilo per minute. So the horse isn't at the top, but we know that they're really quite spectacular. And one of the big hypotheses has always been that the bigger the horse's heart, the faster they run. And there's some pretty nice examples of that on this slide. On this slide here, there is a correlation. We have here in the black dots just some regular or average racehorses in 1987 and their cardiac output on the y-axis versus their heart size in kilos. And then at the top here, we have known heart sizes and cardiac outputs of some of the elite racehorses. And sadly, Secretariat's heart was never actually weighed, but there's a prediction of what the heart says the heart size was. And so, you know, it would be lovely. You can look at this a little bit further and my slide, there we go, my slides are advancing now. And there's additional evidence to support that there is a correlation between the size of the heart and the horse's VO2 max. So in the black dots here, we have national hunt horses. This is a study from the UK. The white dots are flat horses, and then the triangles are just horses that were owned by the previous Animal Health Trust. And in the national hunt horses, there was a correlation between heart size and VO2 max, and so you would assume performance as well. But we all know that heart size alone is not sufficient to predict whether a horse is going to go on to win the Triple Crown, and really it's much more complicated than just that. So over the years, we've selectively bred these horses to have incredible athletic potential and probably increase their heart size as well. And this is one of the major advantages of selectively breeding animals for athletic potential, is there are a lot less damaging structural diseases that we see. So it's really very rare in the horse for us to see dilated cardiomyopathy or hypertrophic cardiomyopathy, or even some of the localized cardiomyopathies that we see in humans and other animal species. VSDs do occur, but they're definitely not common. And although we do see aortic valvular regurgitation in a lot of adult horses sort of later in life, it's quite unusual for that to be sort of a developmental disease or a disease that we see in very early life. And the horses that do develop significant aortic or other valvular regurgitation at a young age typically have a poor performance or are less able to perform than horses that do not. But with selective breeding, we reduce the genetic diversity, and we do increase the risk of having some problems. We know that there's a pretty high prevalence of exercise-induced pulmonary hemorrhage, which is massively underestimated just by the clinical signs of having a horse bleeding out through its nose. We know that a lot of horses have valvular incompetence, so at least 20% of healthy thoroughbreds have some degree of valvular regurgitation when you examine them. And we don't know the clinical significance. We assume in a lot of horses that it's insignificant, but we could follow up on this more. And then we see an increased risk of developing arrhythmias. So this is a human ECG of someone in normal sinus rhythm going into ventricular fibrillation, which is typically fatal unless you can intervene early on. So with that, we'll talk a little bit about arrhythmias and what we know so far. So just a brief review, we typically can recognize arrhythmias sometimes when we're doing cardiac auscultation and the heart doesn't sound nice and regular. And then ideally we would do an ECG to determine what sort of arrhythmia we're evaluating, because with the exception of sort of second-degree atrioventricular block, there are very few arrhythmias that you can definitively diagnose just on auscultation alone. So when we look at an ECG, we look at the RR interval, which is the depolarization of the ventricles, and we can see that it's nice and regular here. But we're also looking for multiple different waveforms, which I'm not going to talk about, but we'll talk a little bit about the P wave, which is important because we get a P and then we get this QRS complex, and this is a nice normal sinus rhythm. However, when you take the horse at the bottom here, you can see that although we do have QRST, we have this sort of undulating baseline. And the QRS complexes are not evenly spaced. So there's a couple here, then there's a bit of a gap, another couple, a bit of a gap. And this is what we would call an irregular rhythm. And this is an example of atrial fibrillation, which I will talk about in the couple of slides. So arrhythmias are really have a surprisingly high prevalence in racehorses, with almost half of them having some kind of arrhythmia at exercise. So when we look at the arrhythmias, we're looking at supraventricular, which originate above the atrioventricular node here. So they're mainly pertaining to the atria, or ventricular, which involves the rest of the depolarization cycle and the ventricles themselves. And you can see that almost half of standardbreds have some kind of supraventricular arrhythmia, and about 30% of thoroughbreds have some kind of ventricular arrhythmia at exercise or in the immediately post-exercise recovery period. And what's even more remarkable is that 16% of apparently healthy racehorses develop complex arrhythmias. Now, these are arrhythmias that if you were a human, you would typically experience, and you experience them under medical care. You may even require defibrillation to correct them. But a lot of these horses seem to be able to spontaneously go back to a normal sinus rhythm without intervention, which is really quite remarkable. So the problem is we don't really know what the relevance of these arrhythmias are. Given how prevalent they are, a lot of them may well not be having an effect on performance. But we know that some, in particular atrial fibrillation, leads to poor performance. And then there's pretty strong evidence that some of these lead to sudden cardiac death as well. But what we don't know is, of the almost 50% of horses that have arrhythmias, how many of them will go on to have significant effects such as sudden cardiac death. And I don't have time to talk about the numerous causes of cardiac arrhythmias in racehorses. The ones that we're most interested in are lone arrhythmias, where they're not caused by underlying metabolic disease or drugs. The horse is fit and healthy. Their hearts are structurally normal, and for whatever reason they go into an arrhythmia, maybe come back out of an arrhythmia, or they go on to collapse and die or have problems. And so I mentioned earlier on that atrial fibrillation is a cause of poor performance, and it's the most common pathologic arrhythmia in horses. And I showed you the ECG before, so we have a normal horse at the top, and then we have this irregular rhythm at the bottom. And we know that horses with poor performance actually have a slightly higher prevalence of atrial fibrillation than regular horses. So the prevalence of regular horses is about 0.11%. But when you look at just horses with poor performance, their prevalence is somewhere between 1 and 2%. So we know that atrial fibrillation is having an impact, and we know that horses that are required to maximally exert themselves, so thoroughbred racehorses, standardbred racehorses, horses that are doing 3-day eventing, are likely to have poor performance with this arrhythmia. What's tremendously difficult is to know when horses are in that rhythm or not. So many horses spontaneously convert. So over 95% of thoroughbred racehorses will go back into a normal sinus rhythm within 24 hours of them having the poor performance episode. But it's very difficult to know whether those horses will go on to have another episode of arrhythmia or whether they will just carry on. And that's certainly one of the biggest questions I get asked at the racetrack is, okay, this horse has atrial fibrillation, they treat it however they want to. What's going to happen? Is this horse going to have it again? Should they sell it, or should they keep running with it and try to, or is there anything they can do to try and prevent it happening? Because obviously they don't want the horse to not run well or to have more devastating effects, such as sudden death. So we have these key questions. So they're kind of twofold. Which horses will develop an arrhythmia, and then which arrhythmias are important. And so what I'm going to talk about is how we use ECG predictions and genetics to go on figure out which horses will develop an arrhythmia. And then in the long run, this is going to help us to determine which arrhythmias are important, whether some of the other more common arrhythmias are contributing to poor performance, whether they're having any clinical disease, and whether or not they're leading to sudden cardiac death, which is a prospective sort of long-term study that we will participate in. So we've already done some research looking at possible ideas for monitoring racehorses on the track. This little device that you can see us holding here is called an Alive Core ECG, and they are very easy to use. We didn't clip the horses, and these were teaching horses that we used that were somewhat hairy, not excessively hairy. And we were able to compare it to sort of the gold standard, which is the televet, and show that they were usable to identify horses, at least in healthy horses that had sort of normal arrhythmias to second degree AV block, and those that didn't, and they were pretty equivalent to the televet for the basic measurements. They're very affordable and so could be used by track vets for horses that need increased monitoring, but they can only be used at rest. They can't be used at exercise because they need to be held in place. So the televet is pretty much the gold standard. It's relatively easy to use. It's not inexpensive, but it's you can use it multiple times. It's not a single-use device, and we can use this before exercise, during exercise, and then in the immediate post-exercise recovery period. And typically, we see the arrhythmias either at exercise or in the post-exercise recovery period. Certainly if we saw some of the supraventricular and ventricular arrhythmias in the resting horses, then we would let the trainers know, and they might not want to train their horses that day. But the challenge with exercising ECGs is they're time consuming. You can only really do them on the days where the horses are being trained hard for what we're looking for. They require both trainer and jockey cooperation. And there's also pretty significant movement artifacts. The electrodes go on underneath the surcingle, and then in standardbreds they go under the harness, and in thoroughbreds they go underneath the saddle. But we still have problems with leads popping off sometimes, and certainly with the horses galloping, there's some movement artifact as well. Sorry, there we go. The next thing to do, I mentioned we're sort of interested in predominantly lone arrhythmias, is ruling out primary structural heart disease using an echocardiogram. And structural changes would be relatively easy to monitor. It would just require fairly regular echoes, but most of the horses with arrhythmias have structurally normal hearts. And so it's something which makes it even more difficult to predict which horses will go on to develop an arrhythmia, because when they're standing in the stall, their hearts look normal, they sound normal, their ECGs are normal. How do you know which horse is going to go on and develop an arrhythmia? And both Molly and I are trained geneticists, and that's one of the things. This is actually one of the studies that first highlighted the possibilities of looking at the genetics of arrhythmias in racehorses. So the atrial fibrillation, which, as I mentioned, is the most common pathologic arrhythmia, is about 30% heritable in horses and humans, which is what we would consider a moderately heritable trait. And what this means is that the horse's genetics contribute about 30% of the risk of an individual going on to develop atrial fibrillation. But the environment is pretty important, and the environment also plays a role as to whether or not the horse is even identified as having atrial fibrillation. We can see some horses that are relatively low level and don't get worked hard, where atrial fibrillation is just picked up at the yearly vaccinations, and you have no idea how long that horse has been in atrial fibrillation because it hasn't been having poor performance. Whereas if you're a racehorse and your atria aren't functioning properly, you're going to be identified much quicker. But what really excited me was that several of the exercise-associated arrhythmias and familial atrial fibrillation are highly heritable in humans. And what that means is that genetics play a really big role. So over sort of 60 to 70% of the risk of these arrhythmias comes from genetics. And that's sort of where we started and where we started reading a bit more about this study. So as I mentioned, these are our key questions, and now I'll go on to how our study is going to help address and answer some of these questions for us. So we have funding through several agencies, including the Grayson Jockey Club, to perform phenotyping on 1,000 racehorses, including auscultation, ECG, echocardiography. And then we're also collecting an additional 100 horses with atrial fibrillation through our collaborators. But all of these horses will have an exercising ECG performed. And first of all, I'm going to talk about the genetics. So having built off the reading that some of the familial forms of atrial fibrillation were highly heritable, Molly had already collected some atrial fibrillation cases. And so during my PhD, we performed whole genome sequencing of these horses to look at all of the DNA base pairs across the genome. And then to prioritize them, we've initially started just at looking in human biologic candidate genes, and specifically ion channelopathy genes. So these are genes that are involved in depolarization and repolarization of the horse's heart. And then we looked at variants that were present in the general horse population, which is a whole genome sequence of 534 horses at an allele frequency of less than 5%. And the reason we did this is because most of the highly heritable disease-causing variants are not common in the general population. And this is data from humans, and certainly is one of the things that we're also looking at what the appropriate cutoff is in horses. But for right now, this is where we're at, because we're trying to find the most highly heritable ones, because that's a great place for us to start. And then we can build on them as we learn more about what the variants are doing. So we also looked at variants that were at the same site as known arrhythmia-causing variants in humans, and we have about 111 of those. And then we have 314 that are within these genes, but not at a known human site. So that gives us 425 variants for follow-up, and they will be genotyped in all 1,100 horses of the study population that we have. And so what are we hoping to find? So if we assume that this sequence here is the reference, and the reference horses, which is actually a thoroughbred, is a G at this position here. But we have a C to G base pair change in one of the important ion channel genes. And when we look at the normal horses, they all have 2 copies of the reference G allele. But when we look at the horses that have atrial fibrillation or one of the other arrhythmias, almost all of them are homozygous, so they have 2 copies for this mutation, and that would suggest that this variant segregates with the phenotype of atrial fibrillation. So we would need to look more thoroughly at this, but this would give us pretty strong evidence to support this variant potentially contributing to the arrhythmia. But as ever, nothing is ever simple. And so there's some horses, this one and this one, that are actually heterozygous for the mutation, which would suggest that if this was a recessive trait where you need 2 copies of the mutation to cause the disease, that these 2 horses should be over here. What's likely happening is these horses have an additional risk mutation that's contributing to them having atrial fibrillation, and that's why we can answer a lot of questions early on, but this is going to need to be an ongoing study until we can get all of the genetic contribution to the disease. So why are we looking for the genetic variants. So genetic testing is incredibly useful for identifying horses that are at risk of developing a particular phenotype. And in this case, we're interested in horses that will go on to collect, to develop an arrhythmia. So if we take a foal here and we collect blood to isolate DNA, we can look at the base pairs that we're interested in that cause the disease. So let's just say it's the C to G here, which this horse is this foal would be heterozygous for. But if it was homozygous, then we would know that it's at a higher risk of having an arrhythmia. Whereas if it was lower risk, we wouldn't necessarily need to monitor it more closely. But if it's a high risk one, we would want to do increased screening, so more regular ECGs, potentially even ECGs the day before, the day of racing, to make sure that the horse is in a nice normal sinus rhythm. And I'll talk about some of the additional ECG monitoring tools that we'll have in the next few slides. And then this will also help us to make educated breeding decisions. We're not going to be in the sort of position to be able to say, okay, this horse has this variant, we should never breed this horse again, because by doing that, we're potentially limiting the genetic diversity of the population even further, and thoroughbreds in particular are incredibly highly inbred, and we don't want to reduce the breeding pool any further. But we could say that that horse with a mutation is not allowed to breed with another horse that has the mutation, because you massively increase your risk of having offspring that have that mutation. Whereas if you breed it to one that doesn't have the mutation, you can decrease your genetic risk while maintaining the available genetic variation as well. And so then moving on to ECG predictions, and this is some really exciting work that there we go, my slide's not advancing, that we started collaborating with the University of Surrey and Rossdale's on. So all credit here has to go to Dr. Jeevaratnam and Dr. Celia Ma. And they've developed a technique that takes resting ECGs, and then uses a computational algorithm which converts the ECG, so each P wave, each QRS complex, and each T wave into a binary code, which the computer then uses. And they've shown very nicely that they can use this to predict horses that have previously had atrial fibrillation. So this here is some complexity analysis on the y-axis, and the numbers aren't particularly important other than to know they're sort of decreasing from 0.03 down to minus 0.03. And in blue here we have control horses, and in red we have horses that have previously had atrial fibrillation but have either been treated and gone back into sinus rhythm, or have spontaneously converted back into sinus rhythm. And you can see here that there's reduced complexity in these horses. And what's really interesting is this is actually similar to some work that they've shown in humans, that human individuals that have previously had an arrhythmia have abnormalities that can be detected at rest. And we're going to extend this, and this is the proposal that was recently funded through the Grayson Jockey Club Foundation, excuse me, to look at other arrhythmias as well. So when they use the feature detection in the computer, it actually doesn't involve any measure of the atrial depolarization or repolarization. It's purely based on the ventricular complexes, and so we think that we'll be able to use this to identify other horses that have other types of arrhythmia at exercise. And what is incredibly helpful by this is that it requires resting ECGs. So obviously for our analysis, we need to do both resting and exercising ECGs. But in the long term, this would mean that we could just do an at-rest trace of an ECG, and then use the computational tools to determine whether or not they're going to be at higher risk of developing an arrhythmia or not. So this is a really viable option for monitoring horses in the future to see which ones are at a higher risk. And what we'll do is determine sort of in the like over their racing periods, are there times where we can use this to say, well, you're even at even higher risk of developing an arrhythmia or poor performance and should therefore be rested or evaluated by a veterinarian. And so sort of the ECGs and complete workups on the 1100 racehorses are going to be able to help us to identify which horses develop arrhythmias and be able to predict this using a combination of their genetics and resting ECGs. And then it's also going to allow us to prospectively follow up high-risk horses and use their performance data to determine which arrhythmias are important for poor performance, clinical disease, and sudden cardiac death. And at the moment, it's really difficult to be able to decide which horses that need monitoring more closely to answer these questions. So our research is going to give us the tools to be able to monitor horses more closely and to figure out what is contributing to their risk of sudden cardiac death. So with that in mind, I'm now going to move on to what we know about sudden cardiac death and how our research is going to impact that as well. So fortunately, sudden cardiac death isn't common, but as we all know, it's absolutely devastating when it occurs, and it's somewhere between 1.1 to 1.5 or so, depending on the year, per 1,000 race starts in thoroughbreds. What that doesn't account for is horses that die on the track in between racing starts. These are only horses that are actually training or racing that are included, and so that we're probably underestimating to some extent this the number of sudden deaths that happen each year. And in thoroughbred racehorses, about 19% of these are classed as sudden death. And what that means is that they die for some reason that's not immediately clear, and they're certainly not related to fractures, catastrophic fractures or catastrophic injuries. So when we look at those, about 19% of the sudden death cases are caused by cardiac abnormalities that are identified at post-mortem. But 47% of them, so almost half of them, have no diagnosis at necropsy. And we just don't know how many of those are caused by arrhythmias. And in humans, the percentages are actually relatively similar. And a large number of the ones that have no diagnosis at necropsy or are termed autopsy negative go on to have some form of genome sequencing, whether it's just candidate gene sequencing or sequencing the whole genome. And about a third of those get a diagnosis at that level. And this adds extra weight to the fact that it's quite probable that genetics are contributing to the risk of arrhythmias in horses as well. So as I've mentioned, arrhythmias are usually undetectable at necropsy. Usually by the time the horse is evaluated, if it's not quite dead on the track, the arrhythmia is already in a terminal arrhythmia, sorry. And so it's not particularly helpful to know what type of arrhythmia is going on. So it's a really big issue of determining how many arrhythmias and which arrhythmias do lead to sudden cardiac death. And so similar to the key questions about the arrhythmias, we also have key questions about sudden cardiac death. So how often do arrhythmias lead to sudden cardiac death, and why do certain horses with arrhythmias collapse and die? And then as we move forward, what can we do to stop these arrhythmias of either developing or prevent them developing from an arrhythmia into sudden cardiac death? So our original study population are focused predominantly on the arrhythmias section, but we're also in the process of adding 100 horses with sudden cardiac death collected across the US from various different racetracks. And our research is going to lead to answering these questions through improved identification of horses at risk of arrhythmias, which will mean that we can monitor those ones more closely to determine when and if they go on to develop sudden cardiac death. We'll be able to monitor them more closely through the combination of using the ECG algorithm and also through sort of encouraging and highlighting the importance of cardiac monitoring in these high-risk horses. And then, as I mentioned before, we'll be able to prospectively look at identifying risk factors for sudden cardiac death. So if we can monitor these horses that are at increased genetic risk or have signs on their ECGs that suggest that they should be monitored more closely, then we can over time determine what is going on in these horses and why does some of them collapse and die. And then this is a huge project, and we're incredibly grateful for everybody that's involved and all the funding that's supported us. Our big goal is to reduce the rate of sudden cardiac death. And so we're studying the basic ECGs and genetics on the 1,000 racehorses, plus the additional 100 standardbred racehorses with atrial fibrillation. And then in this collaboration with Celia and Camelan, we're looking at advanced ECG analysis to determine if we can predict using resting ECGs which horses will go on to develop arrhythmias at exercise. And then we're also planning on echoing all of these horses to look at what structural abnormalities are there, how many of these horses have regurgitation, if they have any flow irregularities or contraction abnormalities, and then we'll look at their performance overall and see whether we can find any correlations or associations there. And overall, this will help us answer our questions and then contribute to this goal of reducing sudden cardiac death. And so before I pass over to Molly, I just want to thank everyone that's involved in this project. It's been from 6 standardbred racehorses with whole genome sequence. It's developed quite spectacularly, and we're incredibly grateful for the funding that we have and our collaborators and co-investigators. And just to highlight just a few, over the last summer, Dr. Gold, Adam and Kendall all helped me collect samples at the tracks, and then also the Stronach group and Dr. Lynn Hovda at the Minnesota Racing Commission, who have been instrumental in helping us get access to these horses and encouraging the trainers to work with us, and obviously the trainers, owners and racehorses themselves, who we couldn't do this work without. And with that, I can pass over to Molly.
Yeah, and I think you'll have to hit refresh on your Google slides because it didn't put the last slide in, but thanks, Sean, for that. And I just wanted to come back in the end. Obviously, this is Sean's baby, and she's done a lot of work on this, but really to just reiterate how grateful we are for the funding, and then to talk about which Sean has already sort of reviewed. And unfortunately, the picture I made for you is technology is limiting that, but just to reiterate, like our overall goals here, when we're done with this project in the end, what we would like to see is that there's a genetic test that allows us, before horses ever go into training or racing, to identify horses that may be at higher risk for what could be fatal cardiac arrhythmias. And then be able to use that information to identify horses that racetrack veterinarians should be looking at more frequently. We know that the best way to do that is with ECGs at exercise, but of course, as Sean pointed out, that's not easy to do, which is why the tools that are funded by Grayson are so important. We would like to be able to put in the hands of racetrack veterinarians something like the iPhone-based ECG that Sean talked about, where then they could essentially have an app that would take what looks like a normal resting ECG that actually, when we look at it computationally, predicts those horses that may have trouble. And knowing a horse may have trouble on a particular day would allow then that veterinarian to decide whether or not the horse should be exercised, or, at fast exercise, or whether the horse should be rested or exercised less intensively, and certainly whether or not the horse should race. In the ideal world, this work, none of it will make a horse not potentially be an athlete, but it will allow us to look at them and really monitor them on the day of racing or exercise. And then also over time, as we start to follow these horses forward, one of the things that we're looking for, will there be key things that tell us maybe this horse should retire because they're increasing risk with age? So with that, I'll say thank you, and we can open it up to questions.
Yeah, so thanks again, ladies, doctors, so much for your time this afternoon, the fascinating research that's being done. As we all know, it's an important issue that we need to tackle in all horses. So for those of you on the webinar, there is a Q&A button at the bottom if you have some questions. But to start it off, I'll ask, and that is, a general question that we got was, how does heart rate, is that related at all to this? Because the horse has such a magnificent heart. What's that first, the heart rate, or how's that related?
So that's a really good question. We know that when horses develop very, very high heart rates, so heart rates over 220 at exercise, they're much more likely to be having a problem. That's abnormal. So to some extent we know that, but I think we'll know a lot more about that when we've got all of these ECGs done. This is, as far as I'm aware, the largest study of actual exercising ECGs. So we'll be able to sort of quantify how high the heart rates are going and whether or not they're whether or not it's just the heart rate and the horse is in a normal rhythm, and that's causing the high heart rate, or whether or not they're actually in an arrhythmia, which is consistent with them having a very high heart rate. So I think that's a great question, and we'll be able to answer it a lot more. Right now, like I say, we know that very high heart rates are a sign of a problem, but we'll hopefully know more by the end of the study as to which ones sort of seem to be more pathologic than the others.
Yeah, and I know that your research is focused on the racehorses because you have Canterbury in your backyard, and that's kind of a captive audience for you guys. And then the racing industry is at one location, whether it's standardbreds, thoroughbreds. But you mentioned an iPhone app is kind of the ideal thing, and I would assume that would be, an average horse owner like myself could go out in the backyard or before an event and talk about how sport horse people would use that as well.
Yeah, and we'd love to. I actually come from a much more sports horse background than a racehorse background, so we would love to work more with the sports horses. But like you say, we're very lucky with the locations of the tracks that are near us. So I think these. Obviously, the genetics we're looking at is predominantly thoroughbreds and standardbreds. There are a lot of thoroughbred event horses. So what we would need to do is extend out whether or not the sort of genetics are similar across breeds, whether or not the warm blood crosses or the warm bloods also have the same variants. And then we can monitor them more closely. And I mean, realistically, we could sort of envision some kind of app where you could take the ECG on the farm, and you're absolutely right, you could use this device at home, record it, and then the app would analyze it and say your horse is having problems or it's not having problems or whatever else, and then you should have a more thorough cardiac workup or something like that. So I think there's a great potential to look at this as well. And you can extend this out to looking at the horses before they go and do their cross-country course. They're probably not going to collapse during their dressage, but they can be monitored if there are any concerns. Those apps could be used. A horse just feels a bit off. Let's just pop an ECG on and just double check if that's the problem. Obviously, there are 20 other things that could be causing the problem, but from the collapsing and dying from a cardiac event issue, that's certainly something that could be useful as well.
Yeah. And the app is nice. That's the same technology that now, right, lets an ECG trace happen on your Apple Watch. You can actually go out and get an alive core ECG to use with your phone. The piece that Grayson's allowing us really to do is to figure out the computational part to say, even if it looked normal, this horse is at high risk, which is pretty cool.
Yeah, so it's not, I think what I'm hearing is it's not only important for you guys to understand population studies, but it's going to get real important to understand individuals as well. And we have to do this research on the population to drill down to the individuals. Is that?
Yeah, exactly. Technology has failed us, but we made a slide to sort of point that out, right? We would be able to, at birth, identify those individuals that we have to monitor differently.
Yeah, so we go from, there's a two-pronged test to it as well, pro or TAC. First is the genetic aspect of it, and then after the horse has been born is in performance. It's the monitoring of that function of the heart, right?
Right. And, people ask the question sometimes, well, why not just do extra monitoring on all of them? That's not always feasible, especially in the track, right? So if we can say to the regulatory veterinarians, these are, this 20% of horses, you need to make sure that you run an ECG before every race. That sort of helps make it more feasible, I think, to really put into practice.
Yeah, and I think that, as you were saying, people are getting more familiar with iPhone apps and heart rates and monitoring their own fitness on whatever band they're using these days. So it's becoming more commonplace. But we don't have any more questions, but I would just like to thank you ladies for, oh, here you go.
Ron found the picture. There it is.
Sorry, it's like I'm going to exit and reiterate the key takeaway points.
Yeah, so the idea that we were just talking about, right, was you take this horse, you decide if he looks like he's going to be normal or not, and then you make monitoring decisions from there.
And poor performance, probably the first factor in all of this for checking in, right, for horse owners, is not performing like itself.
Yeah, I think that's an area that we really are probably missing quite a lot. So obviously when a horse doesn't run well or it doesn't train well, they're very quick to be scoped. They'll look at them from a lameness point of view and everything else. And some of the horses are not. There's nothing wrong with them, but are they having any arrhythmia or not? We had a 2-year-old that we looked at because it wasn't really wasn't performing very well at all, and she was in atrial fibrillation, and fortunately the vet put their stethoscope on and said and we were there, so I looked at it, and she was in atrial fibrillation. But I think that's obviously the most obvious thing, and that's super easy. That's relatively easy to detect. What's takes more effort is these ones that are happening at exercise, because you just don't know. Because by the time they've come back to the stall and you pop your stethoscope on, they're probably back to normal again. I mean, most of the thoroughbreds, the way Canterbury's set up, it's not that far from most of the barns from the track to the barns, but by the time they're getting back to the barns, their heart rate's already dropped down to below 60 and is coming down to 40, despite the fact it could well have been at 180, 200 when they were training. And so if we didn't have the ECG on at the time as the heart rate's going back down again, we'd completely miss any of that information as the horse is going from sort of the sympathetic tone that it needs to race versus the sort of more vaguely parasympathetic tone that they have at rest. So so we're missing all of that. But exercising ECGs are difficult. You can't just, I mean, there are devices you can just put on the racehorses, but a lot of people don't use those because they're expensive and they require interpretation. So if we can combine the genetics and then using the ECGs to screen the horses that do need to be monitored more closely, then hopefully we can get to the point where just an at-risk ECG could be used to say, yes, this horse is at risk. No, it isn't.
Yeah, so the horse has to get up to a certain speed or a certain exercise level to experience arrhythmia, right? Is that?
Yes and no. That to increase our likelihood of getting horses that develop arrhythmias exercise, we're trying to get them when they're breezing or when they're galloping or training for the standardbreds. But probably there are horses that are having arrhythmias at lower levels as well. But we just want to maximize the number that we get. But the more we go on, the more we will know about, how fast do they need to be going to develop an arrhythmia or not. And I think probably the ones that are going to develop arrhythmias are probably developing them at lower speeds as well. We just don't know. So that's more of a hypothesis than anything else.
Well, I don't have any further questions and I don't see any more from the webinar. So again, I'd just like to thank you all so much for not only coming on today and explaining your research, but taking the time and for all you do for horses. It's a great undertaking up there at Minnesota, and we really appreciate your efforts. As everyone knows, this is a problem that has continued to bother us, and we need to, or continue to bother us and our horses, more importantly, and we need to address this going forward. So thank you so much with your research.
Well, thank you very much for your support. I mean, this literally couldn't be possible without it, and we're really excited about this, and hopefully we'll be able to fill in some gaps in the space in the next year or so.
Well, good. We look forward to checking back in with you guys later in the year. Maybe.
Yeah, thank you again.
Yeah. Thank you all, and thanks everyone for attending today. We really appreciate it. Have a great afternoon.
And you.